Ariel. Yes.
Okay, so when these signals come in, what happens? 6 ATP, that would be a nice multiple choice question. How many ATP is needed for tyrosine kinase to become, the receptor tyrosine kinase to become active? 6 of them. So these are some of, I guess, in biology you don't have to memorize that many numbers, but sometimes you do. Okay, sometimes you do. There are 6 ATP that are turned into ADP, and then all of these tyrosines, all 6 of them, are attached to phosphate. Because ATP has 3-phosphate, T, tri, and D, di. Right? Am I making some sense? D, di, T, tri. So they give off one of their phosphate to these tyrosines, and now that tyrosine kinase is active. When it's active, then you have these enzymes that are these protein molecules that are inside of the cell. They come and attach to the tyrosine kinase, and then they become active. Those enzymes inside of molecule inside of the cell, they become active, and that's what the function of tyrosine kinase is. The end product is same as a G protein. If I go back on the PowerPoints, you will see the G protein at the end. At the end, they're supposed to activate some protein molecules, and that's what they do. That's what tyrosine kinase does. At the end, of course, they activate some protein molecules. That's the end product. That's what I'm trying to get at. But how they go about it is different. Okay? So here they are, receptor tyrosine kinase protein, inactive monomers, and then what happens, the dimers, they come together, and then of course they become activated tyrosine kinase regions, unphosphorylated dimers, and fully activated receptors, tyrosine kinase, they become phosphorylated and become active. And after they become active, activate relay protein. Have you guys run relay, in track and field? They give you the python, you run. You all know what I'm talking about. There is an event I'm not talking about swimming. In swimming, there is a relay too, but they don't give you a python. You just see the guy touches the pool, and then you jump in the pool and you swim. You all know what I'm talking about. The relay in swimming, to me, is different than the relay in track and field. In track and field, you're supposed to get a baton. You're running and you put your hand in the bag, and then that's what this is. That's pretty much what this is. Okay, so they come in here, they relay the protein molecules, and the protein molecules go inside of the cell and do whatever they have to do. Okay. So a ligand gated ion, so the next one, the third one, there are 3 types of receptors on the protein, on the surface of the cell. There are 3 different types. G protein is one of them. Tyrosine kinase is the second one of them. I repeated this 100 times so far. But the third one is the ligand gated ion channels, the ion channels. Receptor acts as a gate. When the receptor changes shape, when the signal molecules bind as a ligand to the receptor, the gate allows specific ions such as sodium, potassium, sodium, calcium to get inside of the ion. Here they are. This gate right now is closed. It's not open. It opens when the ligand comes in. Okay, when the ligand comes in, and of course there are ions floating outside of the cell. And what happens next, when the ligand comes in attached to it, then the protein molecules open up, and the ions get inside of the cell. And of course, the end product is cellular response, just like tyrosine kinase and G protein. The end product is inside of the cell, and something happens, and now the gates close back. Okay, for this one again, you don't need any ATP. You just need the ligand. Okay. What are intracellular receptors? That's what Reno said. There are receptors inside of the cell. Okay, there are receptors inside of the cell, such as hormones, and it's a good example of them. In the cytoplasm or nucleus of target cells, small or hydrophobic chemicals, messengers, can readily cross the membrane and activate receptors. So what happens, even though inside of the cell, you have receptors, but there are some kind of stimulators. I'm refusing to use the term hydrophobic chemicals. It means, what does hydrophobic chemical messengers? He's not using the term ligand anymore. He's using the term hydrophobic chemical messengers. They easily pass through the membrane, and then when they come, those messengers come through the membrane, they go inside of the cell and activate those receptors inside of the cell. That's what that one is saying. I hope I'm making some sense. So it's not like activated channels or ion channels or G protein. It's not like that. Okay, it's just hydrophobic molecules. They pass through the membrane, they go inside of the cell, and stimulates most likely the example of hydrophobic messengers are steroids and thyroid hormones of animals. So those are the molecules that he's giving you an example of them. The thyroid hormones, they go through the membrane easily. One activated hormone receptors complex can act as a transcription factor. You don't know what transcription is. You will. We'll talk about it later, turning specific genes on. But since he said that it will make, let me bring you this model as well. I have to have all of my models in order to be able to talk. What is this model of you guys think? It's a nice twisted ladder. It's a model of DNA. So what does that mean? That DNA is found inside of nucleus, right? I want to make it, this is found inside of nucleus, and a portion of this, let's say from here to here, is a gene, and that gene, it gets turned on. See, the genes inside of our cells, there is a switch for turning them on something we talked about it, and turning them off. Oh, I'm sorry, I did something. It turned on and it turned on. What causes to be turned on and off, here's one of them, steroid hormones. Steroid hormones causes these genes to eventually go through transcription, which I will talk about it later on. Okay, material for exam number 3, we'll talk about transcription. Okay, but anyhow, if any of these does not make sense to you guys, later on when you're reading them at home, you can ask me questions. We are just skimming over this one. I'm not going to details of this one, but later on it will make sense, I'm hoping. But anything else, any of these PowerPoints does not make sense, you can ask me during lab, you can ask me at the beginning of the lecture when we are discussing things. Anytime. You can ask me anytime. Remember, I forgot to say this first day of class. I forgot to say this the first day of class. I'm sorry about that. I'm saying it now. Anytime, any word in this class I use that you have not heard before, you're not familiar with before, ask. In this class, I'm not assuming anything that you're already supposed to know. No. But when you go to zoology or botany, they assume you know this, you know that. They talked about this in Bio 1. Am I making some sense, everybody? But here, any word, any sentence, anything does not make sense, please let me know. Transcription, it means when you're turning on the gene. Pretty much we'll talk about it. And based on this gene is turned on, then RNA is being synthesized. We'll talk about it later. So you don't have to worry about it now. Okay, but everything else you should know. Okay, here they are, hormones. Aldosterone is a hormone that a lot of your adrenal gland makes them. Some of the cells of your body makes this hormone, and then they will go inside of the cell. It's a hydrophobic, it goes inside of the cell. Here's a receptor inside of the cell, receptor protein inside of the cell. And then plasma membrane, it goes through plasma membrane, of course. Hormone receptor complex, and then that hormone receptor complex goes into the nucleus. What do you think this is? They didn't label it here, but what do you think this is? What do you think this is? Huh? Anybody? Nobody? It's a what? Say it loud. I say yes or no, and if I say no, so what? You will not lose your salary, right? You will say, no, that's the wrong answer. You don't get dinner tonight. That will never happen. You don't get lunch today. No. What is this? Anybody? What are these gaps? Do you see them in the back? Brian, do you see it? Maya, do you see that? What are they?
Are they pores?
Nuclear pore, thank you. And you do have it in this model too. Nuclear pore. You have it in that model too, in plant model. So they get inside through the nuclear pore and they go to the DNA and turn on the DNA. That's it. They turn on the DNA, and based on that DNA, RNA is being synthesized, and that's called transcription. Amir, what is transcription? When RNA is synthesized based on DNA. The rest of the story I will not let you know. Later on, we'll talk about the next things. Here they are, those pictures one more time. What are protein phosphorylation and dephosphorylation? Phosphorylation and dephosphorylation are regulating protein activity. Protein kinase transfer phosphate from ATP to protein, a process called phosphorylation, which you told me, who was it? Ariel? It was Ariel who gave us the answer that when a protein molecule have phosphor on them, a lot of activities inside of the cell, a lot of chemical reaction that occurs inside of the cell, the protein molecules have to have phosphor attached to them. Where does phosphor comes from? It comes from ATP. And then when the phosphor goes away, that protein molecules become deactive. You just saw one on the cell membrane. You saw one on cell membrane that was tyrosine, right? GTP, tyrosine kinase, I should have said. Okay, so that's what happens. Many relay molecules in signal transduction pathways or protein kinase creating a phosphorylation cascade. Again, signal transduction. Did I talk about transduction in here? It's just like a chain of event. Oh, yeah, I did talk about it here. Molecule A becomes B, molecule B becomes C, molecule C becomes D. That's what transduction is. Okay, so here they are. That's the transduction. You have signaling molecules come to the receptors, activity relay molecules in active protein becomes activated by, of course, ATP, phosphorylation. That's what Ariel was talking about. Maricela, yes, am I making some sense? Okay, so inactive protein molecules become active protein molecules, and again, that active protein molecules turn this inactive protein molecule to that active protein molecule, and this can go on like a cascade. You all know what cascade is. Okay, just like a phosphorylation cascade event. And at the end, we have this molecule that go in and do transcription, translation. Okay, how does phosphorylation takes place? Protein phosphatase says that's an enzyme. Is that right? Ace at the end of the word, it means that's an enzyme. Remove the phosphates from proteins, a process called dephosphorylation. This phosphorylation and dephosphorylation system acts as a molecular switch, turning activities on and off, down and required. But protein phosphatases removes, itself is like an enzyme. Let's talk about this a little bit. So the molecule A has phosphate attached to it. Protein A has phosphate attached to it. But this molecule, phosphatase protein, ace at the end of the word means enzyme. It turns this molecule to this. So now this molecule, they call it is dephosphorylated. This molecule A now is dephosphorylated. This molecule, it is phosphorylated. That's all I'm saying. So what caused this P to be stripped off? That enzyme. The name of the enzyme is a generic term. This is a generic term for a lot of different enzymes that they do dephosphorylation. So protein phosphatase is an enzyme that causes dephosphorylation, removes the phosphorus from a protein molecule. What are some molecules and ions as second messenger? Many signals pathways involve second messengers. Second messengers are small, non-protein, water-soluble molecules or ions that spreads throughout the cell by diffusion. Second messenger messengers participate in pathways initiated by G proteins and tyrosine kinase receptor. That's RTK stands for receptor tyrosine kinase. Cyclic AMP and calcium ions are common second messengers. So what am I saying? What is a second messenger? That you have a cell, you have the ligand right here, it comes to the cell, attaches to the receptor, and then this caused a G protein, you all know, we talked about this, G protein, and G protein relay the message to a second messenger. That would be your first messenger. And this would be G protein will turn on cyclic AMP. They call it, they show it with small c, capital A, capital M, capital P. And I will hopefully, the PowerPoint comes up and I will talk about that a little bit. If not, it's not significant. But G proteins, it turns on cyclic AMP. And cyclic AMP would be your second messenger. And of course, cyclic AMP is important in people who get cholera. Have you heard of cholera, the diarrhea? People get cyclic AMP is important in that. The bacteria, is it saying next one? Yeah, cyclic AMP is one of the most widely used second messenger. Adenylate cyclase, an enzyme that in plasma will convert ATP to cyclic AMP in response to extracellular signals. So what happens? ATP here can become cyclic AMP, of course, by help of that enzymes that are responsible here. He gave you the name of the enzymes, adenylate cyclase. But anyhow, that's what happens. And here they are, ATP. This is the ribose. This is adenine. You remember adenine, nitrogenous base. You remember ribose. You did draw them for me, and hand it in. You remember? I hope. And here is 3-phosphate. So, and then adenylyl cyclase, the enzyme that I mentioned right here, the adenylyl cyclase turns on, it converts this molecule to cyclic AMP. They call this a cyclic AMP. You have a phosphate, and you have a ribose, and you have adenine, and then of course cyclic AMP can become, by help of that enzyme, can become AMP. I don't know, you know this, in our body what happens, ATP can become ADP, and that ADP can become AMP. Adenosine triphosphate, 2-phosphate, 1-phosphate, monophosphate. So that happens inside of our cell. So what he's saying in here, this PowerPoint is saying that it strips off 2-phosphate before it becomes AMP, it becomes cyclic AMP. I hope I'm making some sense. Here they are, first messenger and second messenger right here. First messenger, I did draw it for you guys on the board a little bit. Signaling molecules such as epinephrine. Epinephrine is same as adrenaline in your body. I don't know, Ariel, you know about adrenaline or not. Adrenaline is a hormone that makes you stay and fight, or you better run. Is that right? If I get into a fight, for example, with Brian, I stay and fight. If I get a fight with Reno, I better run, because Reno beats me up, but Brian, I can beat him up. Is that right? They call it fight. You've heard of that. They call it fight or flight. Fight or flight. Think of Brian and Reno. So a fight, adrenaline, okay. And then what happens during this case, this epinephrine, adrenaline, a few things happen in your body. Your heartbeat goes up. Your sugar molecule starts breaking down because you need energy. Maricela, is that right? Your sugar, you need to break down sugar molecules. So either way, if I want to run away from him, I need a lot of sugar molecules breaking down, so I have a lot of ATP to run. And if I want to fight, I still need a lot of energy. But anyhow, that cannot go on in a body for a long time. Okay, some people, soldiers who are coming back from Iraq and Afghanistan, they are used to that adrenaline in their body. Okay, and that's what PTSD comes to a picture. That's what. So having adrenaline constantly secreted in your body is not healthy. To some level, it is. During exam, when you take an exam or quiz, yes, you have high levels of adrenaline. Okay, but other times in your life, when you're driving, there is some. But anyhow, there are many studies done. You can read up on it. But anyhow, so G proteins, of course, you know about this. It stimulates one of the functions. You said, Amir, can you give us one of the functions of G protein? G protein stimulates adenylyl cyclase, which I gave it to you right here. Adenylyl cyclase turns ATP to cyclic AMP, and cyclic AMP is a second messenger, which is protein kinase A cellular responses occurs here. Understanding the role of cyclic pyrimidine, here is cholera. Read it on your own. I'm not going to go over this. There's a lot of the cholera by bacterium, Vibrio cholerae cause diarrhea, constant secretion, constant floating of, and it's caused by too much of cyclic AMP. Okay, cholera. What else is in this chapter? In the plasmic reticulum, pretty much we are high calcium level, low calcium. Again, inside of the cell, like your muscle cells, you have calcium pump, which pumps the calcium into sarcoplasmic reticulum. Here is calcium pump, again ATP needed, calcium pump for mitochondria, ATP needed. If something needs to go outside of the cell, calcium is needed. Here are different type of, we talked about G proteins causes inositol phosphate. Again, I'm not IP3 gated calcium, so the calcium that is going inside of the cell is needed for inositol phosphate calcium channels. Those are sort of some of the things they are mentioning in here. Some of the functions of GTP, if you were wondering what is the function of GTP, here they are, some of them. Animation, let's see what this one I have to say, all right? Sorry for this.
Receptors that are plasma membrane proteins usually affect the cell through multi-step signal transduction pathways. These pathways allow for amplification of signals and signal coordination and regulation. Signal transduction often involves smaller molecules called second messengers and protein phosphorylation by enzymes called protein kinases. Second messengers are small, non-protein molecules that act as intermediaries in signal transmission. Two important second messengers are cyclic AMP and calcium ions.
Cyclic AMP acts as a second messenger in pathways initiated by both G protein-linked receptors and receptor tyrosine kinases.
The hormone epinephrine acts via cyclic AMP as a second messenger. An epinephrine molecule docks with a receptor protein, which acts through a G protein to activate the enzyme adenylyl cyclase. Adenylyl cyclase converts ATP into cyclic AMP, or CAMP, which quickly diffuses through the cell and triggers further steps in the signaling pathway. In this cell, we show it activating a protein kinase. Calcium ions also act as second messengers in signal transduction pathways.
This time we will show a receptor, tyrosine kinase, but G protein-linked receptors can also initiate release of calcium ions. The signal molecule docks with a receptor and activates an enzyme called phospholipase C. The enzyme cleaves a small molecule called inositol trisphosphate, IP3, from a certain kind of membrane phospholipid. IP3 docks with a calcium channel on the endoplasmic reticulum. The channel opens, releasing calcium ions into the cytoplasm, where they activate proteins that carry out cell responses.
Enzymes called protein kinases are important links in many signal transduction pathways. A protein kinase catalyzes the transfer of phosphate groups from ATP to another protein. More often than not, the activated protein is also a protein kinase, which may act on still another protein kinase. One kinase may activate many molecules of the next type of kinase in the chain, thus amplifying the signal until the last kinase activates many molecules of the protein that carries out the final cellular response.
Okay, so this is like a chain of events happening. A lot of proteins are turning into phosphorylated proteins. Okay. Response cell signaling leads to regulation of transcription of cytoplasmic activities. Yes, again, transcription comes like making RNA. Here is DNA. Making RNA based on DNA is called transcription. Okay, but again, a lot of that will come into picture later on. The cell's response to an extracellular signal is called the output response. And here they are again going over the same material over and over and over. Here is one example of, please read this on your own. One example that this morning you had, I did talk about this. This morning you had pancake, and all of that pancake inside of your cells are turning into glycogen. You remember that from beginning of material from cells, the first exam material. Now all of that glucose molecule that are in your liver and your muscle cells, they have to break down to single glucose molecules right here by this method. This method allows the glycogen right here to break down to single glucose molecules. As we talked about it, receptor binding epinephrine and G proteins coupled receptors, and they come inside of the cell. Again, one just it's like a chain of event, a relay of messages, and then what happens? All of the sugar molecules get phosphorylated, and then of course they break down from glycogen, so you can function for the rest of the day. Okay, this is one of the activities that I talked about. Okay, the last thing on this chapter, what is apoptosis, and give a few examples. Apoptosis is not necrosis. In infants, webbing between digital fingers. When you have infants, some babies are born still with them between the fingers. They're attached by layers of membrane. Okay, and I have a picture of them here. But right before birth, those membrane are being shed away. They die. The cells between them die. And that process is called automatic death. Infected webbing between digital cells, tails of tadpoles. Tadpoles, you know, they have a tail, but the frog does not have that big old tail. Am I making some sense? I don't know if you've seen any pictures of tadpoles or you've seen any tadpoles. So all of these happen by apoptosis. In adult infected pathogens, cancers, these are the differences in necrosis and apoptosis. Necrosis is accidental cell death because there is an infection in the body. That's what necrosis is. Apoptosis is a programmed cell death. Okay, less ordered. This is more ordered, and I give you some, show you some pictures how this is more ordered. Lack of signals in most cases. There are signals in this case in apoptosis. Causes injury, infection, and inflammation, and of course this is a programmed cell death. We do not have any of that. So this is what a picture of apoptosis is. As you can see, a normal cell is becoming fragmented, and all of these vesicles, as you mentioned, they eventually are engulfed and being dead. Here is a cell right here, break down to pieces. Even the DNA breaks down to pieces, and a white blood cells comes in, another cell comes in and eats these pieces and bits. And guess what is inside of the cell that breaks down these fragments of this cell that was destroyed? Huh? Anybody?
Enzymes?
Those enzymes are in what package? You're right, enzymes. There is an organelle that has a lot of enzymes. What is it? The what? Anybody? Quiz question today. What is the organelle inside of the cell that has a lot of enzymes?
Cytoplasm?
No, cytoplasm is the medium of the cell. Is that
Is it the
cytoplasm?
Golgi apparatus?
No, Golgi has enzymes, but it will not digest away the foreign material. Lysosome. Lysosome is the suicide baggage of the cell. Why they call it suicide baggage? If the lysosome breaks open, all of the enzymes are released inside of the cell will die. That's what they call it, suicide bag of the cell. But inside this, diffused with lysosome, and lysosome get rid of it. Okay, nucleus fragmented, called DNA lathering and blebbing of cell fragmentation, apoptosis. Here it is the cells that is dying, undergoing apoptosis. Here is the white blood cells engulfing those. Here is the digits, cell undergoing apoptosis. These are our fingers, and then the membranes between our fingers will disappear, and then eventually we'll have a true normal fingers. Okay, let's take 5 minutes break and then go over chapter 8. Okay, anybody? Let's get 5. So that way